Okay, good morning, everyone. So I hope you enjoyed the lectures last week and the fun continues this week as well. So this week we will start with the lectures of Professor Michael Lynch. So Mike is a biologist. For his PhD, he has worked on topics in ecology and behavioral biology. And currently he is Center Director and Professor at the Biodesign Center for Mechanisms of Evolution at the Arizona State University. So Mike's current research is focused on integrating evolutionary biology with cell biology. And I think that's what these three lectures of his are going to focus on. So we are really looking forward to your lectures. So please note that we will play some pre-recorded lectures of Mike. And at the end of the talks, he will take questions. So please note down your questions and then you can ask them when the talk finishes. So I think we can start.
Process. So the next three lectures will be focused on various aspects of this. So today I'm just going to talk about the what we call the population genetic environment, what are rates of mutation, what we call random genetic drift and recombination, how does that vary among lineages. Then the next class I'll be focusing more on the process of evolution, how evolution proceeds by utilizing variation produced by mutation. Then finally, we'll move into some other aspects of how, at a higher level, how cellular complexity might arise by either non-adaptive or adaptive mechanisms. So let's get started here. These are some quotes I really like a lot. You know, we can make observations of the real world, but trying to understand what goes on in the real world, it's more than just stamp collecting. We need a theoretical framework to understand these things. Even, you know, good cooks know you need a theory of cooking to really make things work with your recipe. So here's just a couple of starting quotes to get us off the ground. One by Einstein: make things as simple as possible, but no simpler. So we try to develop general theoretical framework, almost always mathematically, because that allows us to make more precise statements. But of course, we don't want to have things so complex that there's no hope of looking at our basic formulations and understanding what's going on. I won't be showing you all that much math, but I can guarantee you that almost everything I'll be talking about does have mathematical theory underlying it. People who don't like theory might like this next quote: all models are wrong, some are useful. George Box is a statistician. And what he's trying to say is that, you know, we can't predict the location of every atom in the universe, but for many phenomenon, we can do pretty well up to the first or second decimal place. And that really condenses our thought processes down to getting a pretty good mechanical understanding of what's going on. And then this leads to the third quote here from Niels Bohr, one of our great particle physicists. So this is a warning to empiricists. No theory should fit all the facts, because sometimes the facts are wrong. You know, we've used the wrong way of measuring things, done the wrong experiment, and so on. And then finally, Richard Feynman, who many of you may have heard of, one of our more creative physicists ever: what I cannot build, I cannot understand. So what he's basically saying, if I can't write this down in terms of some theoretical framework, and really what he would like to say is, even if it's mathematical, if I can't put it into words, I don't really understand what's going on. So for evolutionary theory, the basis of the theory we build is based on a field called population genetics. As I said, and have shown you before, we're very interested in what we call the population genetic environment, which dictates what natural selection can and cannot do. This is a very well advanced theory, as advanced as the field of biophysics, for example. And so in my view, if we want to try and come up with an evolutionary explanation for some observation, we really need to keep things doubtful until it can be shown to be theoretically feasible at a population genetic level. There's been no real new population genetic mechanisms discovered in the last century or so, and I don't think there will be. It's just a matter of how natural selection or its absence moves around gene frequencies over time. So the types of evolution we're going to see that can occur in a different species depends critically on the population genetic environment, the mutation rate. What we will see is called the effective population size, numbers of individuals effectively genetically in a population, and the degree of linkage or recombination. And we'll see all three of these dimensions vary by about 3 orders of magnitude. And we'll also see that as a consequence of this variation, even though the same rules, population genetic rules apply, there's a different weighting to different activities. And the kinds of evolution that can occur in small populations like multicellular species, there's certain kinds of evolution that can't occur, that can readily occur in microbes and vice versa. And then finally, I'll be emphasizing this throughout the course, is almost all biologists assume that every aspect of every organism, every aspect of biodiversity is a consequence of natural selection and nothing else. But we need to keep our mind open to the possibility that quite a bit of evolutionary change in different lineages is a result of non-adaptive aspects of evolution. And that includes the origin of cellular complexity. So let's start by trying to demystify and clarify what it is we mean by random genetic drift. This isn't terribly difficult than the coin tossing problem. You have a coin, it's unbiased, it's got heads on one side, tails on the other. And you, for example, like to know what the frequency of heads is. But if you flip the coin only once, well, it's either going to be heads or tails, frequency 1 or 0, you're very far off. But as you flip the coin many, many, many more times, you'll eventually get to 50% heads and tails. And that's due to the noise in each individual coin flipping. So let's think now of a population, very tiny one here to the left. There's just 10 individuals, so they're haploid, and some have the black ball there and some have the white ball. So these are 2 alternative alleles at a genetic locus. You start with a frequency of 50%. And then there's a gamete storm. So all these individuals produce a large number of gametes in the big box below. And then we choose 10 more individuals coming out of there. The environment here can only hold 10 individuals. And when we randomly draw them out of here, we see that what's happened is now 60% of the alleles are of the white ball type. And then we repeat this and repeat this and repeat this. And we can see that the allele frequencies are wandering over time just due to the random nature of sampling gametes. Here we have an unbiased process, so there's no natural selection going on. So on average, we should be at 50%, but eventually what's going to happen is we're going to completely lose one of the types by chance. And this might seem like a rather exaggerated situation on the left. After all, a population of 10 individuals is not long for this world for other reasons. But these fluctuations apply to any population of any size. It's just that the magnitude of the fluctuations is inversely proportional to what we call the genetic effective population size. So here to the right, we have an example starting from 50% of a population size of 25. And eventually you see we've lost one of the alleles. We call that fixation of the alternative allele. And that happened at about 40, 45 generations. Now we increase the population size by a factor of 10, so it's up to 250. You can see there's still fluctuations, but they're smaller than they were before. In fact, on average, they're about 10 times smaller. And then we go to a population size of still another factor of 10, 2,500. There's still these little fluctuations. They're tenfold smaller than the last example given. Eventually, though, this line will wander down to either 0 or 1. We'll have lost one of the alternative alleles. And what's important to realize here is that starting with any allele frequency in any particular population size, this trajectory that you see will be different because of the random sampling across generations. So here's just an example of, I don't know, about 20 or so populations, starting at 50-50, 2 allele frequencies, and letting the populations drift around for about 200 generations. And there's 100 diploid individuals here, and you can see a number of them went to fixation for one allele top, another went to fixation for the other allele down at the bottom. A few are still hanging out, only about 3, but eventually they'll get trapped in one of these boundaries of loss or fixation of an allele. So that's random genetic drift without selection. And you can think about it, as I said, just like a coin tossing problem. But what's important to realize here is that what we call the genetic effective population size is not the same as the census number of individuals in the population. Here's one example, a real world example of looking at genetic drift. So in the early 1950s, one of our great population geneticist, Sewell Wright, tortured one of his graduate students named Buri. And Buri set up 256 replicate populations of fruit flies. And this was before the DNA sequencing days, but he had a color marker for eyes, and he started up these populations. So he said 256 of them, and they all had initially 50% of each of the two alleles. So there, in these populations, there were 16 flies. And every generation we pick 8 males and 8 females. If you have 16 flies, they're diploid. So there's 33 states of allele frequency, anywhere from 0 to 16 times 2, 32. Now he started with 16 of each type at this red line here in the middle. And then even after one generation, this is just the frequency distribution of frequencies in the 256 lines. And you can see they've already spread out in terms of allele frequency. And then we're on to generation 5, and they've really spread out. One is completely lost, one of the alleles here. And then we go on and on. You can see this distribution becomes flatter. And by generation 15, and certainly by 19, you see that we've gone from this single peak to a situation where the distribution is U-shaped. And what's happened here, and it continues to happen, eventually we'll lose all these intermediate states, and every population will be fixed for the red allele or the white allele in this particular population. So that's what we mean by random genetic drift. It fits the theory quite nicely. But here's one thing to think about. I won't go through where this math came from, but you can think about the average number of heterozygotes in the population over time. And the number of heterozygotes is going to decline over time on average, because once you fix for one type or the other, you can't have heterozygotes in the population. There's only one allele. But we have this nice theory that projects the decline in frequency of the number or the frequency of heterozygotes over time. And if there's 16 individuals, the prediction is that you would follow this upper line here. But the real data are these dots here. And so what this is telling us is we're losing variation faster than the theory predicts. This is an example. Is the theory wrong, the data wrong, or how do we explain this? And the way we explain this is, so the population is behaving. Here's another line here. It's behaving as though those are only 9 individuals, not 16. And there's lots of reasons this could happen. For example, not all the 16 individuals might actually reproduce. Only 9 of them might reproduce in any generation. That means that the head count is greater than what the population behaves as. 1 or 2 individuals each generation might produce a lot of progeny, and then they dominate the population. And that also reduces what we call the effective size. So the abbreviation for effective size here we'll use throughout is N sub E. Now, this is sort of just a pure ecological view of the problem. And there's lots of ecological, demographic reasons why the effective population size is virtually always smaller than the absolute size. One I just pointed out, if there's variation in gamete production among individuals, especially if this is due to natural selection, some individuals count much more than others. The population behaves smaller than it looks ecologically. If you have population subdivision, so some individuals might be in environments with more nutrients and so on, that will also give you variation in gamete production. Some individuals dominate the scene more than others. If you have an uneven sex ratio, and many species do, in effect, the rarer sex is like a genetic bottleneck, and the population will behave as though it's more similar to the number of individuals in the rare sex than the common sex. And also, if you have a population that fluctuates over time, in the long run, the population will behave effectively as though it's at the bottleneck level, not at the level of the population flush. So these are all ecological reasons, if you like, why effective sizes are always going to be smaller than the number of heads in the population. I'm assuming your organism has a head here. But what's much more important here is it turns out then numbers of individuals is the very nature of our genome architecture. As you all know, all our genes are tied together on a small number of chromosomes in each individual. So genes are linked together physically. And this can do some interesting things when natural selection is operating. So the upper part of this cartoon here, we have something like 9 or 10 chromosomal segments from a population. The little tick marks these would be nucleotide locations where there's variation. You either have the tick, that might be an A, or you don't, that might be a C. So that represents variation in the population. And this blue ball here, this chromosome has picked up an advantageous mutation, so advantageous that after some period of time, natural selection has pulled this chromosome through the population. It goes to fixation over time. We've lost all this other variation in the population in the meantime. So we call that a fixation of a chromosomal segment. And it's happened fast enough here so that recombination couldn't separate other locations on the chromosome from that beneficial mutation. Well, there's two other things that's happened here owing to the fact we've lost all these other background chromosomes. These tick marks over here happened to be another beneficial mutation, but it wasn't as good as the blue ball here. So this chromosomal segment wins, and when it wins, there's a beneficial mutation that's been lost from the population. It's been purged by this selective sweep. And then there's this tick mark next to the blue ball here. That's actually a slightly deleterious mutation, but it's not as bad as the blue ball is good. And so the whole chromosome segment still gets pulled along. And what's happened here is we had three interesting mutations, but natural selection was not as effective as it might be. We've lost one beneficial, we fixed one deleterious, and we fixed the other blue ball beneficial. If we had free recombination between these, so for example, if they were on different chromosomes or something, then natural selection could promote each mutation on the basis of its own merits. If the population size was large enough, we'd have this end state over here to the right. The two beneficials would be fixed and the deleterious would have been lost. So the very mere fact that our chromosomes are tied together on chromosomes greatly reduces the efficiency of natural selection. That's one way. And let me just illustrate this actually with some data. Now people can do evolutionary experiments in the laboratory where we can sequence whole genomes of individuals in culture. And this is a set of results from 8 different populations of the budding yeast, illustrated over to the right, one of our great model systems. The individuals doing this population started out with identical populations with no variation whatsoever. And then over time, new mutations would arise in the populations. And by sequencing the whole population periodically, one could follow the allele-frequency trajectories of molecular variants that appeared in the population. So everywhere you see a line starting here, that means a new mutation came in. If it goes all the way to the top, it means it went to fixation. You lost the original allele at that site in the population. And you can see there's a lot of variation among different populations, but what I want to emphasize here is 2 kinds of populations that were set up here. One, asexual populations. The little yeast cells were not allowed to have sex with each other, and another where sexual reproduction was encouraged. And if you look over to the right, where there's a lot of recombination going on, yeast has a lot of recombination, as I'll illustrate later today's talk, relative to other organisms. You can see that many of these allele frequency trajectories are more or less operating independently of each other. Some go to fixation, Some get off the ground, but later poop out, probably because they've been displaced by another mutation and so on. But what's interesting is to look over to the left. Here, there's no recombination going on. And this links back to the slide that I just shown. What can happen here is that if one new mutation that's beneficial arises in the population, it's going to be in some cell linked to other mutations. And all of those will be pulled to fixation at the same time. So you see these big bundles of mutations going to fixation in tandem, simply because they're all physically linked with each other. So you can really see this phenomenon in experimental populations. So all of that's due to selective sweeps of beneficial mutations, but we have to think about this in a different way as well. And this is not mutually exclusive of what I just talked about, but the vast majority of mutations are not beneficial, they're deleterious. So now let's think about our population of chromosomes here. Again, the tick marks are variant nucleotide sites. And what's happened in this population is that over time, and we'll get some more quantitative information on this, some of these chromosomal segments picked up these deleterious mutations illustrated by the little green ball. Now, if there's no recombination going on, and these are deleterious enough, those chromosomal segments with the little green balls that are deleterious mutations will be eliminated. They'll be purged from the population by negative selection, and will only be left with these chromosomal types without deleterious mutations. So again, selection against deleterious link mutations results in a loss of variation from the population over time. The population is behaving genetically as though it's much smaller than it actually is. Again, purely due to linkage problems in the genome. Now, one key issue that we want to understand at this point is how drift relates to the efficiency of natural selection. And so the way to think about it is look over to the right. Again, each of these balls represents a variant, a genetic variant of a particular nucleotide site. In this case, there happen to be 3 flavors. And let's say this little red one here was a new mutation that just arose. And it will ultimately have 2 fates. One is it might be just lost in the next generation by chance, or it might get lucky due to drift or due to promotion by selection. And you can see going through different time epochs here, eventually this did really well and it went to fixation. So in the meantime, we lost the yellow allele and the green allele. We went to fixation for the red allele. That may have been due to natural selection. The key thing to think about here is how strong natural selection is to random genetic drift, which is the noise in the evolutionary process. The main point is selection pushes things forward in a deterministic way. But if the noise is strong enough, the background noise is strong enough, it will just overcome the deterministic effect of selection. So I don't know, imagine you were driving through a complete blizzard from, let's say, Missoula to Billings, Montana or something. And, you know, if the skies were clear, you'd know how to, presumably you'd know how to follow a map and get to where you want. But now it's a complete whiteout. And you're just driving around, wandering around, and you'll never get to Billings. Or even if you did get to Billings, you wouldn't know it because you were overcome with the blizzard and you just keep driving on to New Mexico or something. So that's the general logic here. Tomoka Oda taught us about the concept of what we call effective neutrality. And other people had thought about this, but I think it's fair to say she was most influential in pointing this key benchmark out. And the point here is that even non-neutral mutations will behave as though they're neutral and completely succumb to the process of random genetic drift if the background noise is sufficiently large. The way to think about this is illustrated here. Let's suppose we have 2 alleles, the big A allele and the little a allele. And these are their fitnesses. We just always set one of the fitnesses equal to 1 because everything depends on relative fitness. So little a, if S is positive, has some small advantage over big A. And you can think about the selective advantage as sort of the little push, the nudge that natural selection would give this little allele each generation. Well, if that nudge in frequency, which is roughly deterministic, if that's overcome by the background noise, natural selection won't be effective. And the background noise in random genetic drift is equal to the inverse of the effective population size. And what that means is what we're asking, if we have a new mutation, if its effect is much smaller, I put a 2 here, that's for diploid organisms, there's no 2 here for a haploid. The strength of selection is an order of magnitude or so less than the power of drift, then selection is rendered ineffective. So we call that effective neutrality. And this becomes important because we think a large fraction of mutations in natural populations have very, very small values of S. Now, associated with this is another concept, which we call the probability of fixation. Here, what we're asking is, if we go back here, a new mutation arises, and it's either going to go to fixation like this, or it's going to be lost completely. And so one of the questions we always ask in evolutionary genetics is, what is the probability that a new mutation in a population of a certain size and a certain selective advantage or disadvantage will go to fixation? And a number of people had worked on this, but Muto Kimura really brought it to the forefront using diffusion theory and a number of complex mathematical approaches. Kimura worked with Oda. They were both at the National Institutes of Genetics. Kimura died a few years ago, but Oda is still there working. And I won't go through the math underlying this formula, but this is the probability of fixation of a new mutation. A new mutation arises in one copy, but in a diploid population, there's n individuals, each of which has 2 genes at the locus. So the initial frequency is 1 over 2n. It'd be 1 over n if it were a haploid population. And the main thing to look at this formula is there's 3 things in here. There's the absolute population size. That's what's determining the initial frequency. There's the effective population size, which, as we said before, is typically much smaller than the absolute. And then there's a selective advantage, S, or disadvantage. So if S is positive, it's advantageous, and if it's negative, it's disadvantageous. These vertical lines here, some of you may remember, mean absolute value. And S is a value between, its absolute value is between 0 and 1. So I won't go, and E is the base of natural logarithms, which I hope some of you remember. So I'll just show you how this plots out graphically. You'll notice there's an S times an NE here. So they're convoluted together. And remember that the key benchmark is, if you just flip this inequality around, we want to know, is 2NES less than 1 or greater than 1? So it's a natural combination. You can think about NES as the ratio of the power of selection to the power of random genetic drift. So that's this axis here. As this gets larger or smaller, farther and farther away from 0, we have more effective selection. And what we're plotting here is this equation, probability that a newly arisen mutation will go to fixation relative to the probability if it were neutral. If it were neutral, the probability of fixation is just the initial frequency, because there's nothing pushing you up or pushing you down. So we can sum up in 3 ways. So if we're in this domain of effective neutrality that Tomoko Oda told us about, that means the product of the effective size and S is tiny, much less than 1. So we'd be in this domain here. And you can see then the probability of fixation is near neutrality. This is why we call it effective neutrality. The mutation isn't necessarily neutral, but the population size is small enough that it behaves as though it's neutral. At another extreme, we can imagine a highly deleterious mutation. We're way over here now. Selection is very effective, and the probability of fixation is essentially 0. So strongly deleterious mutations never fix in populations that they're sufficiently large. But then the other interesting thing here is this: what about a mutation that's strongly advantageous? You might think, well, if it's strongly advantageous, sure, it's just going to go to fixation, no question. But it turns out that that's not true. the probability of fixation of the advantageous mutation is simply equal to twice its selective advantage times this depression term here, which is the effective size over the absolute size. So this means that the probability of fixation of a beneficial allele is never more than twice its advantage. And the reason for this is even a beneficial allele that initially arises is still undergoing a lot of dancing around in noise. And suppose one parent picks up the beneficial mutation and produces 2 kids. Well, there's a high probability neither of the kids will inherit the beneficial mutation, and it's lost. And now we have to wait for the next one to come in. So this is a very useful thing to keep in mind, that not all beneficial mutations go to fixation. In fact, only a tiny fraction of them do. Now let's bring these theoretical ideas home and think about real data. So what I've plotted here is we have measures of genetic effective population sizes for, I don't know, about 30 or 40 organisms in a really convincing way. And this is a summary of this information. We're plotting effective population size of an organism versus the size of the organism itself, the adult dry weight. There's about almost 20 orders of magnitude going along this axis here. We're spanning from bacteria to unicellular eukaryotes all the way up to vertebrates. And I regret to inform you that we humans are over here. And in general, even though there's a lot of us today, historically, there have not been a lot of us. So multicellular organisms have much lower effective sizes than unicellular. And the slope on a log-log plot is about -0.2. That means for every 5 orders of magnitude increase in organism size, there's a 1 order of magnitude decline in the effective population size. So now let's think about this in the context of effective neutrality. So if you live down here, say your effective size is 10 to the 4th, the power of drift is the reciprocal of that, 10 to the -4. So that means that any mutation, if you're way down here, any mutation with an effect smaller than 10 to the -4 is free to fix, because natural selection is just overwhelmed by random genetic drift. So there's a lot of mutations that won't be available to natural selection, and even a lot of deleterious mutations that selection can't eliminate from the population. And then let's move up to the top. This is as good as it gets. So for no organism do we have a known effective population size bigger than about 1 billion individuals. And if any of you who work with microbes know just 1 billion bacterial cells in one pile would barely be able to see. So again, this really emphasizes the genetic effective population sizes many, many orders of magnitude smaller than the absolute number. But what's happening up here, now the power of drift is 1 over 10 to the 9. This means that all mutations with absolute effects greater than 10 to the -9 are visible to selection. So this means there's 5 advantage of in evolution that we big clunky vertebrates and land plants cannot take advantage of. Another way to think about this, if random genetic drift isn't going to play a role in evolution, and one of 2 things must happen. All mutations would have to have effects smaller than 10 to the -9, because they would be so small that no organism could have natural selection seeing them. That would be a really bad situation, because all deleterious mutations just accumulate in any organism. Or the effects would have to be greater than 10 to the -4, in which case any organism could take advantage of them. So this is a big question in evolutionary genetics. What is the distribution of mutational effects in different lineages? So I'm gonna, let me just 2 more slides and then we'll take a break and see if there's any questions. So the way to think about this again in a cartoon fashion is illustrated here. Suppose we have some molecular trait or cellular trait that's under the same form of selection in every organism. Maybe it's ATP synthase or the ability of a ribosome to accurately translate messenger RNAs and something, but what we have is different populations of different sizes. As we move to the right, population sizes are increasing, and selection's relentlessly pushing us up, trying to improve the trait. there's this upper limit that's always going to be there. We can call that the biophysical limit. We're not allowed to break the laws of biophysics. So selection is always trying to get us up here. And as we move to the left, we're getting to smaller and smaller population sizes. So drift is becoming more and more powerful force, and it stops the forward movement due to selection at an earlier point than it does over here. So this is what we call the drift barrier. It's the point at which the rate at which selection pushes you forward is balanced by the noise in the evolutionary process and also by back mutation. So another way to think about this here is I've color coded these populations, blue to red, small to large population sizes. And if you could follow a population through time, it would be wandering around this drift barrier, would sometimes get a little bit above, but natural selection can't keep you there, sometimes get a little bit below, and selection can push you back up. And so if you were to say monitor the evolution of your trait every few thousand generations, you would get some kind of distribution that would look like this. In other words, there's drift around the drift barrier. But as we get to larger and larger population sizes, these distributions shift to the right, and we're now approaching the level of molecular perfection. So we expect population mean phenotypes to evolve to different levels just due to random genetic drift differences alone. So we call this the drift barrier, and there's many examples in biology of drift having an effect on the mean phenotype. One example would be the evolution of senescence. So all organisms age, and we don't really know in a molecular sense why this occurs, but Hamilton, W.D. Hamilton, had a very simple, elegant idea many years ago. And that is that imagine you're an organism and you're reproducing kids for a number of years, and eventually you're not reproducing any longer. At that point, any mutant allele that influences your ability to survive, assuming you're done with parental care and so on, any mutation that influences very late in life and only late in life will just naturally accumulate. So this is a passive mechanism by which senescence evolves in all organisms, and it's essentially a drift barrier, the inability of natural selection to grab onto alleles that are beneficial very, very late in life. There's many other examples. So people who do protein biochemistry, protein folding, and the binding interface between different proteins have typically found that most proteins have stabilities that are right on the edge. So just a 1 or 2 amino acid change is enough to really break down stability or ability of binding interfaces to exist between different proteins. They're not well protected from problems, I guess is what I'm saying. And people have also been interested in the catalytic efficiency of enzymes. At one point, it was enzymes are very good, and at one point, some people felt that they're essentially reached the level of perfection. But if you view the level of perfection of an enzyme catalytic capacity, it's just being the diffusion limit, the rate at which you encounter substrate particles. Most enzymes have catalytic capacities that are orders of magnitude below that. And then the remaining part of the lecture today, I'll illustrate 2 other ways in which the drift barrier applies to the population genetic environment. We're going to see that mutation rates increase in organisms, reduced effective population sizes due to the reduction in efficiency of selection. We'll learn later that most genomes of big multicellular species have experienced a tremendous expansion of excess DNA due to transposable element insertions and so on that can't be countered by natural selection as they can be in bacteria. And then later on in the course, we'll talk about how growth rate can scale with the size of an organism and its effective population size. So I think this brings me to where I wanted to take a quick break and see if there's any questions.
Kavita, have you paused for a reason? We should continue.
Yes, ma'am. Just will play it back.
Yeah.
In a minute.
The population, genetic environment, the power of random genetic drift. And I want to finish up by talking about the two other dimensions, mutation rates and recombination rates. So as I said, we know that most mutations are deleterious. So for that reason, we expect in general natural selection to drive mutation. So let me share screen again. So we just covered the first dimension of the population, genetic environment, the power of random genetic drift. And I want to finish up by talking about the two other dimensions, mutation rates and recombination rates. So as I said, we know that most mutations are deleterious. So for that reason, we expect in general natural selection to drive mutation rates down so that organisms don't deteriorate. That's not to say occasionally a mutation produces a beneficial mutation. It certainly does, but the vast majority of mutations are deleterious. And we can tell that by just growing organisms in the lab, releasing them from natural selection. They almost always go downhill. What we're going to see now is that mutation rates, we just saw that effective population sizes vary by about about 5 orders of magnitude, we said, from 10 to the 4th to 10 to the 9th. Mutation rates evolve to be inversely proportional to the effective population size. Rates range from 10 to the minus 11 in some microbes to 10 to the minus 8 in some vertebrates. So I regret to inform you, but the human mutation rate is the highest one we know, 10 to the minus 8 per nucleotide site. All of you were born with something on the order of 100 new mutations in your genome that your parents didn't have, but provided you through their gametes. We're also going to see a nice lesson about looking at evolution of mutation rates right down to the level of molecules involved with DNA polymerases. And we're going to see that DNA polymerases that aren't used very frequently in cells, except for DNA repair, have very high error rates. And that's consistent with the drift area hypothesis. So the way we think about this, and remember, this is related to lecture number 2, where we talked about the selective disadvantage of a mutator allele. Here I'm looking at it in a somewhat different way. Imagine we have a population size of 10 to the 5th individuals here. And we'll just make these a clonal population. So different individuals have different mutation rates, ranging from high, red, low, purple. And then you can get mutations for the mutation rate. So these different mutation rate classes can change over time. So here we start with a population with a very high error rate. so high that natural selection can really discriminate between individuals with variable mutation rates, and it moves the mutation rate down over time into this gray area, and then we stay there forevermore. Here we start in this gray area and we stay there forever. Now here we start out with a pretty good situation from replication fidelity's perspective, a low mutation rate. The problem here is the selective advantage of this anti-mutator isn't great enough for natural selection to keep you here, and you drift back up into this gray area. So the point here is this gray area, that's the relative bounds of the drift area, and we're sort of stuck there forevermore. You can wander around a little bit, as I pointed out before, but you're stuck in this general area. It's sort of an absorbing area for the trait. Now here, same graph as above, but what we have is a change in scale. We have multiplied the effective population size by a factor of 100, and same stuff goes on, but more slowly because the mutation rate evolves to a lower level, but you still get to an equilibrium mutation rate, but that rate is 10-fold lower than it is here. So the simplest view of the problem here is that mutation rates will evolve to be inversely proportional to the size of the effective size of the population. And over the past few years, we've been able to get really good information on mutation rates. In the old days, this wasn't easy to do. You had to use so-called reporter constructs and so on, like color markers. But now we get this information by sequencing entire genomes in what's called a mutation accumulation experiment. We start with one individual. This is a paramecium, and we feed him or her really well. A day later, we have 50 different lines, let's say. We've only illustrated 6 here. But then we take all these lines through single cell bottlenecks, day after day after day. And by going through a single cell bottleneck, that shuts down natural selection, because if the mutation is in that, and they're asexual, because a mutation arises, it's stuck in the lineage forevermore. And each of these is undergoing independent accumulation of mutations. At the end of the experiment, we isolate the individuals, sequence their whole genomes, and you can count up the number of mutations, divide by the number of nucleotide sites in the genome, the number of generations. You have a measure of the mutation rate. This is just a quick example of such a study with the bacterium called Bacillus. Bacteria have circular chromosomes, so there's 50 lines of Bacillus here. These are just this ring diagram, and each little black dot means a mutation in one of the different 50 lines. In this experiment, after 5,000 generations, there were about 450 mutations found. That's the size of the genome. This gives you a mutation rate about 3 × 10⁻10. So mutation rates are really, really quite low. That's why so much effort has to go in this. We're trying to sample a very, very rare set of rare events enough times to get a good estimate of the overall mutation rate. Another example here, this is another bacterium. It's got a very tiny genome. It's called Mesoplasma florum. It's a bacterium that we know nothing about except that it was isolated off the petal of a lemon tree in Florida. But now we know its mutation rate, and it's almost 100 times higher than it is in Bacillus subtilis, 10⁻8. That's about the rate at which the human, this is dividing every few hours, but it's got a mutation rate per generation about equal to the human mutation rate per human generation. This kind of work has been done now on about 40 or so different bacterial species, a lot of eukaryotes ranging from different kinds of unicellular eukaryotes, yeast, slime mold, Chlamydomonas, and some model organisms as well that are multicellular. And then we want to put this in historical context. 30 years ago, Jan Drake raised the interesting idea that the base substitution mutation rate per nucleotide site was inversely related to the genome size in microbial organisms. This has a slope of -1. This is a log-log plot. And with a slope of -1, this means that the product of the X and the Y axis is constant. And that constant was a 0.003, which some of us started calling a Drake. And what Jan argued was that about 3 in 1,000 baby microbes, no matter what the species, are born with one new mutation. The remaining 0.997 would be mutation free. And Jain didn't really know what was going on, but thought this was a universal description of mutation rates across organisms. And he had this great quote: because this rate is uniform in such diverse organisms, it's likely to be determined by deep general forces. Well, we think those deep general forces are nothing but more than the laws of population genetics. And now with this work repeated in many organisms at the DNA level, Drake's work was all done by these reporter constructs, sort of color marker things, and there was a lot of extrapolation done. But if you look at microbes today with the more modern approach for DNA sequencing, you still get a pattern that has a slope roughly minus 0.1. So that made Jain pretty happy. The problem is when you now look at mutation rates in multicellular species, so invertebrates, vertebrates, and land plants, they're up here. They're not even running in the same direction as predicted by Drake's rule or Drake's law. And as I said, I regret to inform you, but we humans, that other point there is for chimpanzee, we have the highest known mutation rates per nucleotide site out there. So what's going on? Can we unify these results in any way? Well, we don't think mutations are simply causally related to genome size. Just adding more nucleotides to your genome doesn't automatically mean that replication fidelity goes down. Remember, what the drift barrier hypothesis predicts is there should be an inverse relationship between population size, would be on this axis, and the mutation rate across the genome and organism. And it so happens as you move up here, we're talking about organisms with very small long-term effective population sizes. So when we change the plot here, so the x-axis is the genetic effective size of the population, now everything's running in the same direction. The results aren't completely unified. This is the mutation rate per nucleotide site. One of the things you can see, for example, is that bacteria in blue have higher mutation rates than unicellular eukaryotes in green, given the same effective population size. So this is one of the few things unicellular eukaryotes seem to do better than bacteria. But this is also consistent with the drift barrier hypothesis because unicellular eukaryotes have much larger genomes. There's more ways to break the genome than in bacteria. And remember, natural selection operates on the genome-wide deleterious mutation. So to accommodate this, the mutation rates here are predicted to be lower. And we can unify everything by now. It's the same graph as before on the x-axis, but what we've done on the y-axis is multiply the mutation rate per base times the number of bases in the genome that are expected to be under natural selection. And so now you can see that in bacteria, these numbers get down around the region Jan Drake was talking about, 10 to the minus 3. But when you get into big mammalian genomes, including the human genome, the number gets on the order of 10 to the 0 or 1. And what this is telling us from the standpoint of humans, as the average newborn human is picking up one deleterious mutation that wasn't there before in the parents. So 100 total, maybe roughly 1 or 2 are deleterious, and the others aren't having a great effect on fitness. A couple points to make here is the mutation rate is the only trait for which we have a comprehensive theory for the evolution of mean phenotypes across the tree of life, explained, I think, by the drift barrier hypothesis. And one other point to make here is there's often an argument made that there's the way traits evolve is there's a trade-off between speed and efficiency. If you want to move faster in life or carry out reactions at a higher rate, the cost will be an increase in accuracy. But here we see down here, these are microbes that they're reproducing at maximum rates, and yet they're the ones with the minimal error rates. So they're the least error prone. So this goes against the grain of this speed versus efficiency trade-off. One other thing I want to mention with respect to mutation rates is there's mutations kind of rise in different ways in the genome. So when we copy DNA, I think most of you know, we use a molecule called DNA polymerase. So it tries to put a T across from an A and a C across from a G and so on, Watson-Crick base pairs. But occasionally a mistake is made in the polymerization step. So instead of putting a T in here, the cell put a G in here. And there are mechanisms, most of our polymerases have a proofreading domain. And so you can have a secondary check, or eliminate the G and add to a T. But the proofreading step in DNA replication is much less accurate than the first step. And then there's still a third step called mismatch repair that some of you will know about. Now the proofreading missed this mistake and the GA remains as a bubble in the DNA. Mismatch repair can go in and find this bubble and replace it with the appropriate base. Mismatch repair has it's great. It improves things 99%, but it has an error rate of about 1%. Whereas back up here for polymerase, the error rate's more down around 10 to the -4, 10 to the -5. So as you progress up the ladder here in error correcting steps, the error rate actually increases. The cumulative effect, of course, is really quite good. You may not know that most of our genomes have multiple polymerase enzymes in them. So the human genome at this point, there's something like a dozen different DNA polymerases known. There's the housekeeping ones, delta and epsilon. These are the ones that do almost all the replication of DNA. But occasionally there's a problem, there's a lesion in the DNA. And when replication encounters one of those, it elicits the use of another polymerase, one of these guys out here, beta, kappa, eta, and so on. These things have enormous error rates. So you can see this enzyme here, I guess it's iota, has an error rate of 10 to the -1. So out of every 10 bases it incorporates, it makes one error. These polymerases are essential because they plow through certain kinds of DNA lesions that the housekeeping polymerases can't do, but they're also utilized very, very infrequently. Many fewer nucleotide transactions. So we expect them to naturally evolve higher error rates simply because selection isn't enforced as in other enzymes. Now, let's finish up now by thinking about the third dimension of the population genetic environment, the evolution of recombination rates. People have been interested in whether recombination rates can be molded by natural selection to improve the response to selection. We've already seen what recombination can do that can be beneficial. Remember our earlier illustrations about these hitchhiking effects and background selection effects. These become greatly reduced if you have a large amount of recombination, because then selection can operate on individual mutations based on their own merits, as opposed to the cumulative merits everybody you're linked to. So that can be good. The other thing is to look down at this cartoon. One thing recombination can do is to create novel genotypes by exploiting mutations that have arisen on different chromosomes, rather than waiting for new mutations to arise one by one by one by one on the same chromosome. So for example, here, we're looking at a situation where you need a combination of 3 mutations to make a beneficial combination. So that would be this chromosome down here. I'm just illustrating with red. That's really good. And what's happened here is we have one chromosome has 2, but not the third, and the other has 2, and they complement each other. When you get recombination, you bring that chromosomal segment together with that, and that gets you this. Of course, the flip side is you make this with this, and it gets you that. So these guys, because we don't have the beneficial combination, nothing's necessarily going on, but we have created a beneficial mutation. So recombination can bring together independent mutations and create a novel combination faster than would otherwise occur by independent mutations. But there's a flip side to this. Suppose we make this triple combination here. And now imagine there's other chromosomes floating around in the population that don't have that combination. And then they recombine. and now we get these two outputs, we've lost the creative benefit from the prior episode of recombination. So just as recombination can create, it can destroy beneficial combinations. And so the combination of these three different effects has created a lot of interest in whether natural selection can promote recombination rates at all. And what I'm going to show you in this next slide is that there actually is a very, very convincing pattern of recombination variation among organisms that can't be explained by advancement of recombination rates by natural selection, but rather seems to be a simple physical consequence of the way we undergo meiosis. What we're looking at here from, I don't know, about 100 or so different organisms ranging from unicellular species, yeast is right here, and up into big multicellular species with large genomes. So the human genome, I guess, is probably one of these points here. Our genome size is about 3 billion. Yeast is down around only roughly 10 million bases and so on. And then some land plants with tens or even hundreds of millions of bases in the genome. This is on the x-axis. The y-axis is the recombination rate per physical distance. It's a log-log plot. So these dashed lines indicate a slope of -1. And what you can see is the data are consistent with this -1 slope relationship. So the larger the genome size, the lower the recombination rate per physical distance. I put two lines in here. Down here, organisms have small numbers of chromosomes. Up here, they have large numbers of chromosomes. So what's going on here? Well, this relationship is sort of biology 101. We don't really know why genetics develop this way, but basically in all organisms during meiosis, there's typically an average of just one crossover event per chromosome arm. That means if you evolve larger chromosomes and retain the same number one crossover per chromosome arm, you reduce the average amount of recombination per physical distance. You double the size of the chromosome, you reduce the amount of recombination by 50%. And the reason we have this vertical distribution here is here, as I said, we have larger numbers of chromosomes. So the genome sizes are the same, but if you subdivide your genome into more and more chromosomes, that will get you more recombination per physical distance. So this is known as the one crossover per chromosome arm rule in genetics. There's not a lot of wiggle room left here to argue for modification of recombination rates by natural selection. If you know the number of chromosomes in a genome and the total genome size, we can predict with about 98+% accuracy the average amount of recombination per physical distance. So the take-home message is out here, large genomes, these are species with small effective population sizes. They have low rates of recombination per physical distance. Yeast, very tiny genome, large effective size. It's the most recombinogenic organism we know of. And this seems to be a simple outcome of this rule, this physical constraint on the process of meiosis. So I'm going to end with one more quick slide and then a summary. You might think of, well, bacteria don't have meiosis, so there's no recombination in bacteria, so we can treat those in the ideal way of no recombination. But in fact, bacteria, some of you know, have taken microbiology, have different ways of recombining genomes with other members of the population. The bottom line is that there's as much recombination if we compare it with the mutation rate in genomes. There's just as much recombination going on in typical bacteria on average as there is in eukaryotic genomes. So we shouldn't think of bacteria as being non-recombinogenic. It's just a different kind of recombination than meiosis. And finally, this consequence of recombination and its influence on genetic variation can be looked at in the following way. There is variation in the amount of recombination along chromosomes within organisms. generally recombination rate is low near centromeres and higher out in the arms of chromosomes. What you can do then is you can look at the amount of variation at different genetic loci in different regions of recombination along the chromosome. And these data are for humans here, about 20 different genes. And you can see that the genes in the area of low recombination have lower amounts of variation. They're succumbing more to these effects of background selection and selective sweep. So this is completely consistent with the theory of how recombination influences variation. So let's sum up what we've learned today. There's a lot of material sort of squeezed into just an hour and 15 minutes, but I hope what we've done today is at least give you an idea of how the forces of population genetics vary among organisms. Population genetic environment itself evolves. If you increase organism size, you cause a reduction in the population size, effective population size, and you cause a reduction in the rate of recombination because chromosomes get bloated with excess DNA. And this leads to an increase in the power of random genetic drift. And that in turn leads to an increase in the evolved mutation rate. So these three aspects of the population genetic environment co-vary in fairly natural ways. And the point here is that depending on where you are in this three-axis domain of variation in the population genetic environment dictates how you can evolve. So mutations, recall that the definition of selective effective neutrality. Mutations with effects smaller 1NE are overwhelmed by genetic drift. So that means small organisms with higher effective populations can use a wider range of mutational effects in adaptive evolution. Larger organisms have a smaller NE. They therefore have a reduced capacity for evolutionary fine tuning. We can't use mutations of small effect, but only of those of very large effect. So the prediction is that evolution will be more coarse-grained in terms of utilization of variation. So we expect, because of this gradient in the effectiveness of selection, at all levels of bioorganization, we expect mean phenotypes to scale just with population size, even in situations where there's exactly the same selective forces going on in different organisms. We expect to see these gradients in the patterns of different phenotypic traits. So that takes us to the end of today, right on time, it looks like. What I can do, since I'm not stuck in a room with another class coming in behind me,
Okay, so I think we can now take questions. Yeah, so Mike is here online. So I saw some raised hands. So go ahead, unmute yourself and ask your questions.
I had one question. I didn't properly understand what you explained about the evolution of senescence being dictated by the drift barrier hypothesis. So could you please explain that once again?
Yeah, I think just realized a few years ago that I think Bill Hamilton was probably the first person to talk about a drift barrier hypothesis without actually using the words. So his idea for the evolution of senescence is really quite simple, and that's if you think about mutations at different genes, assuming there are genes that influence you at early life, midlife, late life, very late life, and that's an open question whether they exist, but assuming they do, then the strength of selection is going to decline with genes expressed later in life, simply because you will have expressed most of your fitness earlier in life. So for example, if you're in your post-reproductive years and you're not engaged in maternal care or grandmaternal care or something, there'd be no selection against deleterious alleles that only have effects late in life. And there's actually a gradient as I just gave you an extreme example, but there's actually a gradient with just increasing age, decline in the strength of selection that would go on. And so he argued that senescence was an inevitable outcome of this simple process, and that it follows at the molecular level, if this hypothesis is true, there would be no real standard generic mechanism at the molecular level to explain senescence. It would be just due to random mutations at different loci that happen to have accumulated in different species.
Thank you.
Does that help?
Yes, yeah, thank you.
Yeah.
Can I follow up on that? I was just curious, can you explain again where the drift component comes in? So then is the prediction that for populations with very low effective population size, we would expect senescence to be much like later? Is that later? Yeah. Whereas for populations with larger effective size, you'd expect to see senescence to not be around as much because they're basically not going, selection is not anyway fixing any beneficial mutations as efficiently.
Yeah, I hadn't thought about that too deeply before, but I think that's a good point. So I think selection is pervasive. There is almost nothing that one could literally call an absolutely neutral mutation. The only thing I could think of is if you went from, say, an AT base to a TA or a GC to a CG, because there's bioenergetic differences between the amount of ATP it costs to even make single nucleotides. So I think selection is operating always. And so I think maybe you're right. If the problem here is that, natural organisms have all kinds of different ecologies, and there's different things that have led to different age-specific rates of reproduction and mortality. But I think you're right. I think the prediction would be that if for some reason if you were lucky enough to have two organisms with identical ecologies, so selection and the life history, the one with the smaller effective population size, I would suggest would have earlier senescence. The selection would be exactly the same, but you'd be hitting the roof of the drift barrier at an early point. So it's an interesting point to think about. I don't know how I would really like to figure out how we can actually test Hamilton's hypothesis. It's really never been tested in a formal sense, but that's the good thought experiment, I think.
Thanks.
Any more questions?
Apologies for the of the pictures of my students over to the right of my slides. We'll try and fix that for the next lecture. I didn't realize those were going to be in there.
I think it was more or less okay, but yeah, if it can
I covered up a few keywords, but
yeah. Okay, so right now I think I
have a question.
go ahead.
Yeah, so I have a question about mutation rates. So do we know if mutation rates are at all correlated with lifespan? Because I would expect that maybe if the lifespan is longer, then you'd have a larger mutation rate because to adapt to fluctuating environments or something. Does that make sense, the question?
well, if there were a correlation, it would be that, I mean, crudely speaking, you would have to say that things with longer lifespans, like us mammals, we have the highest mutation rates relative to microbes, which have much shorter lifespans. We're comparing quite different organisms here. probably the better way to look at it would be to look at it within a group of organisms that have, roughly comparable body plans and so on. But, I don't think well, let me just say that I think a paper will be coming out in maybe the next few months that has done quite a large elaborate study in one particular group that might be helpful here. But in general, I don't think we have enough data to really say that there's a correlation. Remember, the mutation rate we're measuring is the mutation rate per generation. It's from offspring to offspring. And so I think at this point, we can't really say. It is true that that older males in particular, but also older females, transmit a larger number of mutations to their offspring. So just age itself can do this, simply because mutations accumulate in the germline when it's just sitting there. And of course, the male germline, there's repeated cell divisions, and that might be part of it, but there's still a lot of debate about these kinds of things.
Okay, yeah, thank you.
You're welcome.
Okay. Yeah, I think there are no more questions I don't see any. Yeah, so I think we can stop now. So thanks, Mike. So I know it's very late. I apologize. I mean, I somehow couldn't help it, but I hope, it's not too bad, but let's see how this goes.
Yeah, no problem. And it coincides with my next 2 lectures. So it'll be a similar format then.
All right, yeah. So we will try to take care of this in-between break somehow to try to see if it can be made more smooth. Yeah.
Right. Yeah, sorry about that.
Yeah, it's okay. Yeah, fine. Thanks so much, Mike. And see you on Wednesday.
2 days, right? 2 days from now? Okay.
Yeah.
Thanks a lot.